Welcome to the deep dive. Have you ever stopped to wonder how your body does it?
Does what exactly?
Well, you've got trillions of cells, skin cells, muscle cells, brain cells, all doing completely different things.
Right. Specialized
But
functions.
they all have the exact same DNA inside them. How does that work? It's not magic, right?
No, not magic. It's this incredibly sophisticated process called regulation of gene expression in eukaryotes.
Okay, let's unpack this. Today we're doing a deep dive into this biological symphony. We'll explore how your cells decide precisely which genes to switch on or off and when and where. Basically, how they conduct the music.
And it's not just some fascinating biological puzzle, though it definitely is that.
Yeah.
It's really fundamental. Understanding this helps us grasp everything from normal development, how a single fertilized egg becomes a whole person,
Wow, yeah.
to really serious diseases like cancer, where this regulation often goes completely haywire.
So mistakes in this process can have huge consequences.
Absolutely huge. So we'll explore the many, many intricate levels of control, right? From how the DNA itself is packaged up,
Okay.
all the way to the final sort of tweaks made to the proteins after they're built. It's a multi-layered system.
Now, I remember from basic biology that bacteria are much simpler in this regard.
Oh, definitely. Bacterial cells have a pretty streamlined approach. Transcription-making RNA from DNA and translation-making protein from that RNA,
Right.
they happen almost at the same time, right there in the cytoplasm. They're coupled.
But for us eukaryotes, it's totally different.
That's right. What's fascinating here is just how different our cells are structured. Our DNA, it's tucked away inside the nucleus.
Right.
But the ribosomes, the protein factories, they're out in the cytoplasm.
So they're physically separated.
Exactly. That spatial separation and the time delay it creates means transcription and translation are uncoupled.
Okay.
And that automatically creates multiple new points where regulation can happen. Things bacteria just don't deal with.
And our RNA needs more processing too, doesn't it?
It really does. Our messenger RNAs, the mRNAs, they undergo quite extensive processing.
Yeah.
They need a special cap on one end, the 5' cap,
Okay.
and a long tail of A bases on the other, the 3' poly A tail.
Mm.
Plus, these non-coding bits called introns have to be precisely cut out or spliced.
And all that before it even leaves the nucleus.
Correct. And unlike bacterial mRNAs, which often get translated straight away and then break down quickly, eukaryotic mRNAs can hang around for a surprisingly long time.
Mm.
Their half-lives can vary a lot.
So they can be more stable.
Yeah, and they can be shipped off to specific parts of the cell to be translated right where the protein is needed. And then there's a whole other layer, post-translational modifications to the proteins themselves. Way more complex than in bacteria.
Okay, so let's start at the beginning with the DNA itself. You said it's packaged. It's not just loose strands floating around.
Not at all. It's intricately packaged with proteins, mainly histones, into this complex structure called chromatin.
Chromatin.
Yeah.
Right. You mentioned it's like a library.
Yeah, think of it like a massive, super organized library. Access to the specific books, the genes, is really tightly controlled by how they're shelved and stored.
So this packaging controls access. How does the cell manage that? And how does it decide which books to, open up?
Well, this compact structure, what we sometimes call closed chromatin, physically blocks the machinery needed for things like DNA replication, repair, and especially transcription.
So it's off limits by default, mostly.
Pretty much. Before a gene can even be transcribed, its local chromatin structure has to become open. And the nucleus itself isn't just a random bag of chromosomes. It's got architecture.
Architecture. How so?
Each chromosome tends to occupy its own distinct space, a chromosome territory. They stay relatively separate. And the genes that are actively being transcribed are often found near the edges of these territories.
Why there?
It seems they're positioned near these channels, called interchromatin compartments, which probably helps with coordinating their expression, processing the RNA efficiently, and getting the finished mRNA out to the cytoplasm. It's very organized.
Okay, so how does the cell actually open that tightly packed chromatin? What are the specific tricks it uses?
There are several key mechanisms working together. First, histone composition. Most of the little spools, the nucleosomes, use a standard histone called H2A.
Okay.
But sometimes the cell swaps invariant histones, like one called H2AZ. These variants can make the nucleosome less stable.
Ah, so easier to move or disrupt.
Exactly. Less a barrier to transcription. And you often find these H2AZ nucleosomes near the control regions of genes.
Makes sense. What else?
Second, histone modifications. This is a huge area. It involves chemically modifying the histone proteins themselves, usually on their little tails that stick out.
Like adding tags.
Precisely. Adding small chemical groups like acetyl groups or methyl groups or phosphate groups. Acetylation is a big one. Adding acetyl groups tends to neutralize some of the positive charge on histones.
And DNA is negatively charged.
Right, so acetylation reduces the attraction between histones and DNA, loosening things up, promoting that open chromatin state.
And enzymes do this.
Yes, enzymes called histone acetyltransferases, or HATs, add the acetyl groups. And other enzymes, histone deacetylases or HDACs, remove them, which helps close the chromatin back down and silence genes.
So it's reversible, like a switch.
Very much like a switch. And third, there's chromatin remodeling. This is more physical. It involves large protein machines called chromatin remodeling complexes.
What do they do?
They use energy from ATP to literally slide nucleosomes along the DNA or even eject them completely. This physically clears the way for the transcription machinery to access the gene.
Wow. Okay, so histone variants, chemical tags, and physical remodeling. That's quite a toolkit for controlling access. What about the DNA itself? Can it be modified too?
Absolutely. DNA methylation is another really crucial layer, especially in mammals like us.
Methylation. Adding methyl groups.
Yes, adding a methyl group, usually to a cytosine base in the DNA sequence, specifically where a cytosine is followed by a guanine. These are called CpG doublets.
CpG.
And these CpG sites are often clustered together in regions called CpG islands, which are frequently located in or near gene promoters.
And what does methylation do?
The evidence is very strong that DNA methylation generally acts to repress gene expression. It's a silencing mark.
Can you give an example?
Sure. Think about the inactivated X chromosome in female mammals. One of the two X chromosomes is largely shut down.
Right, X inactivation.
Well, that inactive X is typically heavily methylated. And if you experimentally block that methylation, some of those genes can actually turn back on.
So it really locks genes down. How?
It can work in a couple of ways. It might directly block transcription factors from binding to their target DNA sequences, or it can recruit proteins that bring in those chromatin remodeling complexes and HDACs we talked about, the ones that promote closed chromatin.
So it reinforces the silencing. This raises an important question. You mentioned cancer earlier. How critical is understanding this methylation?
Oh, it's incredibly critical. Because abnormal patterns of these modifications, chromatin changes, DNA methylation, are hallmarks of many cancers. For instance, you might find hypermethylation, too much methylation, silencing genes that normally repair DNA, like MLH1 or BRCA1.
And silencing DNA repair genes sounds bad.
It's very bad. It can contribute to the genetic instability that drives cancer growth. So yes, understanding these epigenetic marks like methylation and histone modification is vital for understanding disease and developing new treatments. They are truly master switches.
Okay, so let's assume the chromatin is now open around a gene. The book is accessible. What happens next to actually start transcription to copy the DNA into RNA?
Right. This is transcription initiation.
Yeah.
And it's incredibly precise. It's orchestrated by specific DNA sequences near the gene, called cis-acting elements, and the proteins that recognize and bind to them.
Cis-acting, meaning they're on the same piece of DNA.
Exactly. They're part of the gene's own regulatory landscape. The most fundamental cis-acting element is the promoter.
The promoter, that's where RNA polymerase binds.
That's the core idea, yes. The promoter is the region of DNA directly upstream of the gene's coding sequence. It's recognized and bound by RNA polymerase II, that's the main enzyme for transcribing protein-coding genes, and a suite of other proteins called general transcription factors, or GPFs.
And the promoter tells the polymerase where to start.
Precisely. It specifies the exact transcription start site and which direction to go along the DNA. Now, promoters themselves can vary. Some are focused core promoters.
Focused.
Meaning they have one very specific start site. These are often found in genes whose expression needs to be tightly controlled, turned on or off very precisely.
Okay. And the other type?
The other type are dispersed core promoters. These are more common, actually, maybe in about 70% of our genes. They have multiple weak start sites over a broader region.
And what kind of genes have those?
Often housekeeping genes. Genes that need to be expressed pretty much all the time in most cells at a sort of basal level.
So within these promoters, are there specific DNA sequences, like little codes that the proteins recognize? You mentioned passwords earlier.
That's a great analogy. Yes, focus promoters especially contain various short, conserved sequences called core promoter elements. These are the binding sites for specific regulatory factors.
Like what?
Well, there's the initiator element in R, which actually overlaps the transcription start site itself. Then there's the famous TATA box, usually located about 30 base pairs upstream.
Ah, the Tata box. I've heard of that one.
Yeah, it's a key binding site for one of the first general transcription factors, TFID. There are others too, like the BRE element, MTE, DPE.
Mm.
It's a bit of an alphabet soup.
Okay.
And besides these core elements right at the start site, many genes also have proximal promoter elements a bit further upstream. Things like the CAT box or the GC box.
Yeah, what do they do?
They generally help increase the baseline level of transcription. We know they're important because if you mutate sequences like the CAT box, the rate of transcription can drop dramatically.
Got it. So promoters are right next to the gene. But what about other regulatory elements? You mentioned enhancers, and you said, here's where it gets really interesting. They can be far away.
You're absolutely right. Enhancers are fascinating. Unlike promoters, which have to be right there, adjacent to the gene,
Yeah.
enhancers can be located, well, almost anywhere relative to the gene. On either side, thousands, even millions of base pairs away. Sometimes they're even found within an intron of the gene they regulate.
A million base pairs away. How does that even work?
It's pretty amazing. They're still cis-regulatory. They affect genes on the same chromosome, but their position and even their orientation, whether they're pointing forwards or backwards relative to the gene, often doesn't matter.
But they still boost transcription.
Significantly. That's why they're called enhancers. They can dramatically increase the rate of transcription, often in a very specific way, like only in certain cell types or only at certain times during development.
And is there an opposite? Something that turns genes down from afar?
Yes. There are also silencers. They share that same flexibility in position and orientation as enhancers, but they act as negative regulators, decreasing transcription.
Okay, so enhancers boost, silencers repress, and they can be ages away. How do these elements actually, exert their influence on the gene?
They function primarily as binding sites for proteins called transcription factors. These are the proteins that actually read the regulatory code. We can broadly classify them as activators, which bind to enhancers and increase transcription,
Okay.
and repressors, which bind to silencers and decrease transcription.
And these factors themselves can be regulated.
Highly regulated. An activator might only be produced in, say, liver cells, ensuring a gene is only turned on in the liver, or a factor might only become active when it gets a phosphate group, added phosphorylation, or when it binds to a hormone, like a steroid hormone. It's a really intricate network of a control.
Could you walk us through a real example, show how all these pieces fit together for one specific gene?
Sure, the human metallothionein 2A gene, or MT2A, is a great case study.
Metallothionein sounds like it involves metals.
It does. The protein it makes binds to heavy metals like zinc and cadmium, helping protect cells from their toxic effects. It also helps combat oxidative stress.
Okay, so a protective protein.
Right. And it's expressed at a low basal level in most cells.
Yeah.
But if the cell gets exposed to heavy metals or even certain stress hormones like glucocorticoids, its transcription rate shoots way up.
So it's inducible.
Exactly. And its regulation involves a whole collection of these cis-acting elements we've been discussing.
Yeah.
It has core promoter elements like a TATA box and an initiator. It has proximal elements like GC boxes.
Okay.
And then it has several enhancer elements. There are metal response elements, MREs, and a glucocorticoid response element, GRE, among others.
So specific sites for metal signals and hormone signals.
Precisely.
Yeah.
And different transcription factors bind to these sites. A factor called SP1 helps with mesotranscription. transcription.
Yeah.
Then there's a factor called MTF1 that responds to heavy metals. What's amazing is MTF1 normally hangs out in the cytoplasm. But when heavy metals enter the cell, MTF1 binds them and then moves into the nucleus to activate the MT2A gene via those MREs.
Wow, that's direct sensing.
It is. And similarly, the glucocorticoid receptor binds to stress hormones, then moves into the nucleus and binds to the GRE to activate the gene. So this one gene integrates signals about basal needs, metal stress, and hormonal stress through this complex array of elements and factors.
That really illustrates the complexity. So these factors bind the DNA, maybe far away. How do they actually talk to the RNA polymerase sitting at the promoter? How do they bridge that physical gap?
That's where DNA looping comes in. The DNA is flexible enough that it can actually bend around, bringing distant enhancers or silencers physically close to the promoter region.
Ah, okay. So they make contact despite the linear distance.
Exactly. And researchers have a few models for how this interaction then influences transcription. One is the recruitment model. Yeah, the idea is that activators bound at enhancers, once looped close to the promoter, help recruit the general transcription factors and RNA polymerase II itself,
Hmm.
making the assembly of the whole pre-initiation complex, or PIC, more efficient and stable.
So they help gather the necessary machinery?
Right. Sometimes other proteins called coactivators act like bridges, linking the DNA-bound activators to the factors at the promoter, forming these large complexes called enhanceosomes. Repressors, conversely, could interfere with this recruitment.
Okay, that makes sense. What's another model?
Another model focuses on chromatin alterations. The looping might bring enzymes that modify histones, like those HATs we talked about for activation, or HEACs for repression, right to the promoter, changing the local chromatin structure to either favor or inhibit transcription.
So they modify the local environment.
Correct. And a third idea, the nuclear relocation model, suggests that looping might actually help move the entire gene region to a different neighborhood within the nucleus.
Like moving it to a transcription factory.
Sort of, yes. Moving it to a region that's rich in transcription machinery, making it more likely to be transcribed. Or, conversely, moving it to a repressive area. And importantly, these models aren't mutually exclusive. A single gene might use elements of all three mechanisms. It shows how adaptable the cell is.
Right, it's got multiple ways to achieve that fine control. Okay, so we've transcribed the gene. We have a pre-mRNA molecule. But you said the journey isn't over, especially in eukaryotes. There's processing.
A lot of processing. And this introduces even more layers of regulation.
So what does this all mean for gene expression? What's the biggest regulatory step after transcription?
Well, one of the most impactful post-transcriptional mechanisms is alternative splicing.
Alternative splicing, that's where the introns get removed, right? But
Yeah,
alternative.
During splicing, the introns are removed, and the exons, the coding parts, are stitched together. But for many genes, the cell can choose which exons to include in the final mRNA.
You mean it can skip some exons? Or include different combinations?
Precisely. A single pre-mRNA from one gene can be spliced in multiple different ways, generating different mature mRNAs or splice forms.
And these different splice forms would code for different proteins.
Yes, they encode related but distinct proteins called protein isoforms. Even a small change, like including or excluding one exon, can add or remove a chunk of amino acids, potentially changing the protein's active site, where it goes in the cell, or what other molecules it binds to.
So effectively, one gene can produce a whole menu of related proteins. That's truly remarkable efficiency.
It really is. Think about the AMMIC gene in fruit flies, Drosophila. It codes for a myosin motor protein used in muscles.
Okay.
Well, different muscle types in the fly express different isoforms of this AMMIC protein, generated by alternative splicing. And these isoforms have slightly different contractile properties suited for that specific muscle.
So specialized versions from the same gene?
Exactly. There's even an embryonic isoform that, if you force the fly to express it in its flight muscles later on, it actually slows down the wing beat frequency because its kinetics are different. It clearly shows how splicing generates functional diversity.
Are there common patterns to how this happens?
Yes, there are several types of alternative splicing events. The most common in animals seems to be cassette exons, where a whole exon can either be included or skipped entirely.
Like a tape cassette you pop in or leave out?
Kind of, yeah. We also see alternative splice sites being used within an exon, sometimes intron retention, where an intron is actually left in the final mRNA, which is more common in plants. There are mutually exclusive exons, where choosing one automatically excludes another from a set, and even alternative start sites, promoters or end sites, polydenylation signals, can generate different mRNA versions.
This sounds like it massively expands the protein repertoire from a limited number of genes.
It absolutely does. The human genome, we think, has around 20,000 protein-coding genes. That number hasn't changed much in recent estimates.
Right.
But deep sequencing studies suggest that over 95%, maybe even higher of our genes with multiple exons, undergo alternative splicing.
95%.
Yeah, which means the actual number of different proteins we can make, our proteome, could be vastly larger. Some estimates put it in the hundreds of thousands.
Wow. Is there a really extreme example of this?
The poster child is probably the DSCAM gene in Drosophila again. It's involved in wiring the nervous system.
Okay.
Its mature mRNA has only 24 exons, but the pre-mRNA has multiple alternative choices for 4 of those exon positions. The number of possible combinations is staggering.
How many?
Theoretically, this one DSCAM gene can produce 38,016 different protein isoforms through alternative splicing.
38,000 from one gene. That's insane.
It is. And the thinking is that these slightly different DSCAM proteins act like unique identity tags on the surface of neurons, helping them find their correct partners and avoid wiring mistakes.
Yep.
It provides incredible specificity.
Okay, alternative splicing is clearly crucial. So what happens if it goes wrong? Are there diseases linked to splicing errors?
Yes, definitely. Defects in alternative splicing are now recognized as the cause of a growing number of human diseases, sometimes called spliceopathies.
Spliceopathies. Okay, example.
A really significant example is myotonic dystrophy, or DM. It's a relatively common inherited disorder, affects about 1 in 8,000 people. It causes progressive muscle weakness and stiffness. That's the myotonia, plus cataracts, heart problems, cognitive issues. It's multisystemic.
And that's caused by bad splicing.
Intriguingly, yes, but not directly by a mutation in the protein being spliced incorrectly. There are two main forms, DM1 and DM2, caused by mutations in two different genes. But the mutations themselves are expansions of short repeated DNA sequences.
Repeat expansions, like in Huntington's.
Similar idea, yes. In DM1, it's a CTG repeat, and in DM2, a CCTG repeat. These repeats get transcribed into the RNA. And the problem is that these repeat-containing RNAs accumulate in the cell nucleus.
Okay, so the RNA itself is the problem.
Exactly. These abnormal RNAs act like sticky traps. They physically bind to and sequester crucial RNA-binding proteins, RBPs, that normally regulate the alternative splicing of many other genes.
Ah, so they soak up the splicing regulator.
Precisely, which means those RBPs can't do their job properly on dozens, maybe hundreds of other target mRNAs, particularly ones important for muscle and neuron function. That's why the disease has such diverse symptoms. It's a disruption of many downstream splicing events.
That's a fascinating mechanism, and understanding that must help with finding therapies.
It really is. Current research is focused on things like trying to degrade those toxic repeat RNAs or maybe blocking the RBPs from getting stuck to them.
Okay, so alternative splicing is huge. What about just how long an mRNA molecule lasts? Does that matter for regulation?
Absolutely. The amount of any specific mRNA in the cell at a given moment, what we call its steady-state level, depends on two things: how fast it's being made, transcription rate, and how fast it's being broken down, degradation rate.
Right, supply and demand or supply and disposal.
Exactly. And controlling the degradation rate is a major way to regulate how much protein gets made from that mRNA. mRNA stability can vary hugely. Some last minutes, others can last for days. And this decay is actively regulated.
How are they usually degraded?
The most common pathway in eukaryotes is deadenylation-dependent decay. It starts with enzymes called deadenylases, chewing back that poly-A tail we mentioned earlier.
Shortening a tail. Okay.
Once the tail is short enough, often the 5' cap gets removed by decapping enzymes, and then the main body of the mRNA is rapidly degraded by enzymes called exoribonucleases, like XRN1, munching from the 5' end inwards.
So tail removal, then cap removal, then destruction.
That's the main route. There's also a less common pathway that starts with decapping directly. And again, RNA-binding proteins play a big role here, binding to specific mRNAs to either protect them from decay or target them for faster degradation.
What if an mRNA is just wrong? Like it has a mistake, an error from transcription or splicing. Does the cell have quality control?
Oh yes, eukaryotic cells have very sophisticated mRNA surveillance pathways to deal with faulty messages.
Surveillance, like watching them.
Kind of, yeah. The best studied pathway is called nonsense-mediated decay, or NMD.
Nonsense.
It targets mRNAs that contain a premature stop codon, a stop signal that appears too early in the message before the normal end.
Ah, which would lead to a short, probably useless protein.
Exactly. Making truncated proteins is wasteful and can even be harmful if they interfere with other things. NMD recognizes these premature stop codons, often by their position relative to splice junctions or the poly-A tail, and it triggers the rapid destruction of that faulty mRNA before it can be translated much. It's crucial quality control.
Okay, so mRNAs are processed, checked for errors, and their lifespan is controlled. Does translation always happen right away, or can mRNAs be, like, put on hold or sent somewhere specific first?
Great question. No, it's not always immediate. Some mRNAs are stored for later use, translated only when needed. And even more fascinatingly, many mRNAs are actively transported and localized to specific regions within the cell.
Localized, you mean shipped to a particular corner of the cell?
Precisely. And they are only translated once they arrive at that destination. This allows the cell to generate asymmetric protein distributions, meaning you can have a high concentration of a specific protein in one part of the cell and very little elsewhere. This lets different parts of the cell do different things.
Can you give an example of that?
A classic example is actin mRNA in cells that need to crawl, like fibroblasts, healing a wound, or migrating neurons.
Okay, actin is part of the cytoskeleton, right? for movement.
Exactly. These crawling cells extend a protrusion at their front edge called a lamellipodium, and they do this by rapidly building new actin filaments right there.
So they need actin protein at the leading edge.
Right. And what happens is that the actin mRNA itself is transported and localized specifically to the lamellipodium.
The mRNA gets sent there first.
Yes. This localization depends on a short sequence in the mRNA's tail end, in the 3' untranslated region, UTR, often called a zip code.
A zip code, like for mail.
Uh-huh. Exactly like that. An RNA-binding protein called ZBP1 recognizes and binds to the zip code sequence, probably starting in the nucleus. It escorts the mRNA out to the cytoplasm, but initially it also blocks translation, keeps the ribosomes off.
So it's transported but silenced.
Right. ZBP1 also hooks up with motor proteins that carry the whole mRNA ZBP1 complex along the cytoskeleton tracks towards the leading edge.
Delivering the package.
Exactly. Once it arrives at the lamellipodium, a signaling pathway involving an enzyme called CR kinase gets activated. CRC adds a phosphate group to ZBP1.
Phosphorylation again?
Yep. And that phosphorylation causes ZBP1 to release the mRNA. Now, the ribosomes can jump on, and actin protein is synthesized right where it's needed to drive cell movement.
That is incredibly elegant. Zip codes, transport, localized release. Wow. And problems with this could cause issues.
Definitely. Defects in mRNA localization in neurons, for instance, have been implicated in neurological disorders like Fragile X syndrome. It's vital for proper cell function and development.
Okay, so we've covered mRNA regulation extensively, splicing, stability, surveillance, localization. But what about all the other RNA molecules in the cell, the ones that don't actually code for proteins? Do they get involved in gene regulation?
Oh, absolutely. What's fascinating here is the realization over the last couple of decades of just how important non-coding RNAs, ncRNAs, are. We used to think of RNA mainly as RNA and ribosomes and tRNA carrying amino acids.
Right, the supporting cast for translation.
But now we know there's this huge, diverse world of ncRNAs that act as critical regulators, especially after transcription.
Like what?
A major mechanism is RNA interference, or RNAi. This is where small, non-coding RNA molecules guide the silencing of specific target mRNAs.
Small RNAs silencing bigger RNAs. How do these tiny RNAs work? They sound like incredibly specific cellular tools.
They really are. There are two main types of these small non-coding RNAs, snCRNAs, involved in RNA. First, small interfering RNAs, or siRNAs.
siRNAs, where do they come from?
They're typically processed from longer, double-stranded RNA, dsRNA molecules.
Yeah.
This dsRNA might come from a virus infecting the cell, or from transposons, those jumping genes in our genome. It's often seen as a defense mechanism.
Okay, so the cell chops up foreign or unusual dsRNA.
Exactly. An enzyme called dicer acts like molecular scissors, dicing the dsRNA into short pieces, about 22 nucleotides long, which are the siRNAs.
And then
These siRNAs get loaded into a protein complex called RISC, the RNA-induced silencing complex. RISC contains a key protein from the Argonaute family, which has an activity nicknamed slicer.
Slicer. Sounds ominous.
It is for the target mRNA.
Yeah.
RISC unwinds the siRNA duplex, usually keeps one strand as a single-stranded guide, and then uses that guide sequence to find messenger RNAs in the cytoplasm that have a perfectly complementary sequence.
Perfect match.
And when it finds that perfect match, the slicer component of RISC cuts the mRNA in two, leading to its rapid degradation. So it effectively silences that gene by destroying its message.
Wow. Targeted destruction. What's the other type of small RNA?
The other major players are microRNAs, or miRNAs. These are also short, around 21-23 nucleotides, but unlike siRNAs that often come from external sources, miRNAs are encoded by our own genes.
So our genome makes its own tiny regulators.
Yes, hundreds of them. They start as longer transcripts called primiRNAs that fold into hairpin shapes. An enzyme in the nucleus called drosha processes them into primiRNAs. These get exported to the cytoplasm, where dicer, the same enzyme as for siRNAs, cuts them into the mature double-stranded miRNA.
And then they go into RISC too.
Correct. One strand gets loaded into RSC, again acting as a guide. But miRNAs often work slightly differently than siRNAs, especially in animals.
How so?
Animal miRNAs usually bind to target sites in the 3 untranslated regions of mRNAs called miRNA response elements, MREs. And the match between the miRNA and the MRE is often imperfect, not a perfect complementary sequence.
Partial match. So RISC doesn't slice it.
Usually not. With a partial match, the main effect of miRNA-RSC binding is to block translation of the mRNA. It prevents the ribosomes from doing their job, or sometimes it triggers faster denylation and decay. So it still silences the gene, but often by repression rather than destruction.
So siRNAs usually lead to cutting. MiRNAs often lead to blocking translation?
That's a good general rule, though there's overlap.
Yeah.
And we have over 1,500 known miRNA genes in humans regulating all sorts of processes like cell division, development, brain function. They're incredibly important.
And this whole RNAi discovery, it's been huge for research in medicine, right?
Absolutely revolutionary. It gave us a way to experimentally turn down specific genes to study their function. And it opened the door to RNA-based drugs. In 2018, the first FDA-approved RNAi drug, patisiran, came out. It uses siRNAs delivered in nanoparticles to treat a rare genetic disease by silencing a problematic gene. It's a whole new therapeutic approach.
Amazing. Are there larger non-coding RNAs involved in regulation too? Not just these tiny ones?
Yes, indeed. There's a whole other class called long non-coding RNAs or LNCRNAs.
Long. How long?
The cutoff is pretty arbitrary, but generally they're defined as ncRNAs longer than 200 nucleotides, so significantly longer than miRNAs or siRNAs.
And they don't code for proteins.
Correct. They often look a bit like mRNAs. They can be capped, polyadenylated, even spliced, but they lack the proper start and stop signals to be translated into proteins. And there are thousands of them encoded in our genome, maybe around 17,000 LNCRNAs.
And what do these long ones do?
They have incredibly diverse functions, and we're still figuring a lot of it out. Some LNCRNAs bind to those chromatin modifying complexes we discussed earlier, guiding them to specific genes to turn them on or off.
So they can influence chromatin.
Yes. Others can directly interact with transcription factors, modulating their activity. Some can even regulate alternative splicing by binding near splice sites on pre-mRNAs. A few can even pair up with mRNAs to form double-stranded regions, which might then trigger an siRNA-like response.
Wow, a real jack of all trades.
They seem to be. And one really intriguing idea is that some LNCRNAs act as competing endogenous RNAs, or siRNAs.
Competing? Competing for what?
Competing for microRNAs. The idea is that these siRNAs contain binding sites, MREs, for certain miRNAs within their own sequence.
Oh, okay.
So they act like molecular sponges, soaking up those specific miRNAs and preventing them from binding to their normal mRNA targets.
Ah, so by binding the miRNA, they protect the other mRNAs that miRNA would normally repress.
Exactly. They essentially de-repress the target mRNAs by acting as decoys for the miRNAs. It adds this whole other layer of crosstalk and complex regulation within the cell's RNA networks.
That's mind-bending, sponges soaking up regulators. Okay,
Yeah.
so we've gone from DNA accessibility to transcription to RNA processing stability, localization, and now these non-coding RNA regulators. We must be near the end of the line now. We have a protein.
Almost, but not quite.
Yeah.
Even after a protein is successfully synthesized, the story of gene expression regulation isn't over.
Still more?
Still more. The protein's activity, its stability, where it goes in the cell, who it interacts with, all of this, can be further controlled through post-translational modifications, or PTMs.
Modifying the protein after it's made.
Correct. These are typically covalent additions of different chemical groups to specific amino acid residues in the protein, catalyzed by enzymes.
And what's the most common PTM? You mentioned phosphorylation earlier.
Phosphorylation is indeed the king of PTMs. It's estimated to account for maybe 65%, almost two-thirds, of all such modifications.
Adding phosphate groups.
Right. Enzymes called kinases add phosphate groups, usually to serine, threonine, or tyrosine amino acids. And other enzymes called phosphatases remove them. Our genome codes for hundreds of different kinoses and phosphatases, providing just countless ways to toggle protein function.
And what does adding a phosphate group do?
It usually causes a change in the protein's 3D shape, its conformation. That change can switch an enzyme on or off, alter how well a transcription factor binds DNA, change where the protein is located in the cell, or affect its interactions with other proteins. It's a fundamental regulatory switch.
Okay, phosphorylation is huge. What else happens post-translationally? What about getting rid of proteins when they're old or no longer needed? Is there a system for that?
Absolutely essential. That's where ubiquitin-mediated protein degradation comes in. This is the main pathway eukaryotes use to specifically target proteins for destruction.
Ubiquitin. Sounds ubiquitous.
It is. It's a small, highly conserved 76 amino acid protein found in all eukaryotic cells. And it gets covalently attached to other proteins that are destined for the cellular recycling bin.
Like putting a tag on them.
Exactly like that. The process is called ubiquitination. Often, multiple ubiquitin molecules are added, forming long polyubiquitin chains on the target protein. And these chains act as a degradation signal.
A signal for what? Like a cellular garbage disposal?
Precisely. There's a large protein complex called the proteasome Think of it as a barrel-shaped structure lined with proteases enzymes that chop up proteins. The proteasome recognizes proteins tagged with those polyubiquitin chains. It grabs onto the tagged protein, unfolds it, removes the ubiquitin tags, they get recycled, and then feeds the protein chain into its central chamber where it's chopped into small peptides, effectively destroying it.
So, a highly specific protein shredder.
A very specific and regulated one. The key regulatory step is determining which proteins get tagged with ubiquitin in the first place. That's controlled mainly by a large family of enzymes called ubiquitin ligases.
Ligases, they join things.
Yes, they catalyze the attachment of ubiquitin to specific target proteins. Each ubiquitin ligase typically recognizes only a specific set of target proteins, and we have over 600 different ubiquitin ligase genes in our genome.
Wow, that's a lot of targeting capability.
It is. It's estimated they regulate the stability of maybe 40% of all our proteins. So controlling protein lifespan via ubiquitination and proteasomal degradation is a fundamental and broadly used mechanism for regulating almost every aspect of cell biology.
What an absolutely incredible journey. We've gone from DNA, coiled up tightly in chromatin, all the way through transcription, RNA processing and regulation by non-coding RNAs, translation, and finally modifying and degrading the proteins themselves.
It really covers the whole flow of genetic information and all the control points along the way.
It's just mind-boggling, the sheer number of layers, the complexity, the intricate checks and balances. It's clear that the complexity of life depends on this astonishingly sophisticated multi-level control system. It truly is a symphony.
It really is. And this raises an important question, or maybe just an appreciation. When you think about all those regulatory elements, promoters, enhancers, silencers scattered across the genome,
Yeah.
and the huge cast of transcription factors, activators, repressors, the coactivators, plus the powerful non-coding RNAs, the miRNAs, the LNC RNAs, and then all those post-translational modifications like phosphorylation and ubiquitination.
Woo, it's a lot.
It really underscores the elegance and the incredible precision with which our cells manage this immense library of genetic information.
Right.
Every single cell, every moment is playing out this symphony of finely tuned gene expression. It's what allows for all the specialized cells and functions that make us who we are.
And just scratching the surface, understanding even a fraction of this gives us such powerful insights into health and disease. It's clearly opening doors for amazing future therapies targeting these regulatory pathways.
Definitely. The potential is enormous.
Well, we hope this deep dive covering the key concepts from this essential area of genetics is giving you some incredible aha moments. We hope you have a newfound appreciation for the hidden, intricate dances happening inside your cells right now.
It's a dance that keeps us all going.
Thank you so much for joining us on this deep dive. A special thank you for being part of our last-minute lecture family. Keep learning, keep exploring, and keep being curious.